50 years ago, NASA conducted an experiment on Mars that produced one of the most controversial results in the history of space exploration. The experiment appeared to detect biological activity. Not proof of an alien civilization. Not a Martian creature crawling across the surface. But chemical activity that, according to the experiment's original design, could have been produced by living microorganisms. And then NASA called the results inconclusive. For decades, the Viking missions remained at the center of one of the greatest unanswered questions in science. Did Mars ever contain life? Now, that question is being examined again. In the 1970s, Mars was almost completely unknown. NASA had just finished the Apollo moon landings, and plans for human missions to Mars had been pushed aside. But scientists still wanted answers. So NASA built something extraordinary. 2 spacecraft, Viking 1 and Viking 2, were sent toward Mars. Each carried an orbiter and a lander. The landers were essentially robotic laboratories designed to operate millions of miles from Earth with technology that was primitive by today's standards. Viking 1 launched in August 1975. Viking 2 followed shortly afterward. After months of traveling through space, Viking 1 reached Mars and successfully entered orbit. On July 20, 1976, it became the first American spacecraft to successfully land on the Martian surface. Viking 2 landed several weeks later in a completely different region. For the first time, humanity had 2 robotic laboratories sitting directly on another planet. And one of their most important jobs was to search for life. The Viking landers carried several biological experiments, each designed to look for different signs of microbial activity. One experiment, called the gas exchange experiment, placed Martian soil in a chamber and exposed it to a nutrient solution. Scientists wanted to know whether something in the soil would react with those nutrients and release gases. The experiment produced changes in the gases inside the chamber, including oxygen and carbon dioxide. That result was interesting, but scientists could not immediately determine whether biology or chemistry was responsible. Then came the pyrolytic release experiment. This test exposed Martian soil to carbon dioxide and light and searched for evidence that organisms might incorporate carbon into biological material. Again, the result was interpreted as a possible positive. But the most famous experiment was the labeled release experiment. The principle was remarkably simple. Scientists placed Martian soil inside a chamber and added a nutrient solution containing radioactive carbon. If microorganisms were alive in the soil and consumed those nutrients, they could release radioactive carbon-containing gases. And that is essentially what happened. The labeled release experiment produced a positive response. Even more interesting, when some samples were heated to destroy potential microorganisms, the biological signal disappeared. To Gilbert Levin, one of the scientists who designed the experiment, this was powerful evidence that Viking had detected microbial life. Levin remained convinced for decades that Viking had discovered life on Mars. But NASA had another instrument that complicated the story. It was called the Gas Chromatograph Mass Spectrometer, or GCMS. Its job was to search for organic molecules in Martian soil. At the time, scientists expected that if life was present, organic molecules should also be detectable. The GCMS did not find the organic compounds researchers expected. That created a major contradiction. The biological experiments were producing intriguing results, while the chemical analysis appeared to show no organic material. NASA ultimately concluded that the Viking experiments had not provided definitive evidence of life. The results could potentially be explained by unusual chemistry in Martian soil. And that decision shaped the way Mars was viewed for decades. But then something important happened. NASA kept going back. Curiosity arrived on Mars in 2012. Perseverance arrived later. And modern instruments began detecting organic molecules in Martian rocks and soil. Curiosity found organic compounds in ancient rocks and later detected increasingly complex carbon-containing molecules. These discoveries did not prove that life created them. Organic molecules can form through non-biological chemistry. But they demonstrated something important. Mars was not chemically dead. It contained many of the ingredients associated with the chemistry of life. Then scientists revisited one of the biggest problems with the original Viking experiment. Martian soil contains perchlorates. These chemicals were not fully understood when Viking landed. When heated to the extreme temperatures used by the GCMS, perchlorates can break down and react with organic material. In other words, the very process designed to search for organic molecules may have destroyed some of the evidence before the instrument could detect it. That possibility changed how scientists looked at the Viking results. It did not prove that Viking found life. But it weakened the argument that the negative organic results were enough to rule life out. Then came another major development. Researchers began reanalyzing the original Viking data using modern statistical techniques. Some analyses reported similarities between the Viking measurements and biological datasets from Earth. Some researchers have argued that these results make a biological explanation plausible. Others strongly disagree. And that disagreement is exactly why the Viking mystery remains alive. Science does not work by choosing the most exciting explanation. It works by eliminating alternatives. A positive biological signal is exciting. But if an unusual chemical reaction can produce the same signal, scientists have to consider it. That is the central problem with Viking. The experiments produced results that looked biological. But researchers could not completely eliminate non-biological explanations. And that means the evidence remains controversial. Modern Mars missions have made the mystery even more interesting. Perseverance has been exploring Jezero crater, a location that once contained an ancient lake and river delta. That makes it one of the most promising places on Mars to search for evidence of ancient microbial life. The rover has discovered organic carbon and minerals in rocks that preserve clues about ancient chemical processes. Some rocks contain unusual textures and mineral patterns that researchers have described as potential biosignatures. One particularly famous sample comes from a rock nicknamed Chayava Falls. Scientists identified features that could potentially have formed through chemical reactions associated with microbial activity. But once again, the word that matters is potential. Scientists have not discovered a Martian bacterium. They have not photographed a living organism. They have not brought back a rock and proven beyond reasonable doubt that it contains fossils. Instead, they are finding increasingly complicated evidence that ancient Mars once had environments capable of supporting life. And that distinction is incredibly important. Mars may have once been warmer and wetter. It had rivers. It had lakes. It had groundwater. It had organic chemistry. And it had environments that could have been habitable billions of years ago. The question is whether something actually lived there. There is another reason the Viking results deserve renewed attention. Technology has changed dramatically. The computers aboard Viking were extremely limited compared with modern spacecraft. Today's Mars rovers can perform sophisticated chemical analyses, identify minerals, examine rocks at microscopic scales, and search for organic molecules using instruments that Viking scientists could only dream about. Curiosity uses instruments capable of analyzing the chemical composition of rocks and atmospheric gases. Perseverance carries instruments specifically designed to study rocks and identify potential biosignatures. And unlike Viking, Perseverance is also collecting carefully selected samples for possible return to Earth. That could eventually change everything. A laboratory on Earth can perform analyses that are simply impossible for a small robotic rover millions of miles away. Scientists could examine Martian material with multiple instruments. They could repeat experiments. They could search for isotopic patterns. They could examine microscopic structures. They could test competing chemical explanations. And most importantly, they could preserve the samples for future generations of scientists. That is why Mars sample return remains so important, even though the mission architecture has faced major technical, financial, and scheduling challenges. Because if Mars ever contained life, the evidence may already be sitting inside one of those rocks. There is also a larger issue. The Viking missions were designed specifically to search for active microbial metabolism. Most missions that came afterward were designed primarily to study geology, climate, water, habitability, and organic chemistry. That means humanity has spent decades exploring Mars without repeating the exact kind of biological experiment that created the original controversy. We have become much better at finding the ingredients of life. But we still have not answered whether those ingredients ever came together to create life on Mars. And that is the real mystery. The Viking experiments did not prove that Mars has life. But they also did not prove that Mars is lifeless. Modern discoveries have made the old negative conclusion much harder to treat as the final word.